First, we learn about endospore bacteria. Endospores are formed in response to unfavorable growth conditions in the bacterial environment, most commonly induced by the limitation of nutrients carbon and nitrogen. Endospore production is a very important characteristic of some bacteria, allowing them to resist adverse environmental conditions such as desiccation, chemical exposure, extreme heat, etc. Sporulation, however, is not the first response of the bacterial cell to nutrient depletion. Only if these mechanisms fail to provide enough nutrients for continued vegetative growth, the cell commits to the sporulation pathway. The process of spore formation is divided into distinct morphological and biochemical stages. Endospore formation is a complex process and may be divided into 7 stages. In stage 1, an axial filament of nuclear material forms, followed by an inward folding of the cell membrane to enclose part of the DNA and produce the forespore septum. Stage 2. The membrane continues to grow and engulfs the immature endospore in a second membrane. Stage 3. Next, cortex is laid down in the space between the 2 membranes, and both calcium and dipicolinic acid are accumulated. Stage 4. Protein coats then are formed around the cortex. Stage 5, and maturation of the endospore occurs. Stage 6. Finally, lytic enzymes destroy the sporangium, releasing the spore. Stage 7. Sporulation requires about 10 hours in Bacillus megaterium. The location where an endospore is within a vegetative cell is also useful for distinguishing bacterial species. Endospores may be located in a terminal end of the cell, subterminal near the end of the cell, or central middle of the cell position. A particular species of the genus will form spores in a specific area, producing another useful taxonomic identification tool, and therefore useful in identifying the species of bacteria. Thank you for watching. Like, share, subscribe for more such videos.